Welcome to Pharma Drama, the channel where we look at the science of healthcare and healthcare products. In this video, I'm going to explain distribution coefficients, what they are and how they differ from partition coefficients. So distribute your time between the kitchen so you can get yourself a drink and your computer, and let's make a start. Distribution coefficients are very similar in principle to partition coefficients, so it should come as no surprise that I'm going to suggest very strongly that you watch the video on partition coefficients before watching this one. The link is below, so if you haven't watched it yet, then what are you still doing watching this? Go and watch it and then come back. Okay, assuming you're up to speed, I will remind you that the basis of partitioning is that any compound, whether it is a drug or not, is likely to have some affinity for polar solvents, such as water, and some affinity for non-polar solvents, such as oils and organic phases. Water and organic solvents do not usually mix. We say they are immiscible. So when added together in a container, the organic solvent will float on top of the water. When a material is added to this mixture of solvents, the molecules will naturally distribute between the liquids, with a greater proportion dissolving in the organic phase if the compound is lipophilic, and a greater proportion dissolving in the water if the compound is hydrophilic. The ratio of the concentrations in these two phases is called the partition coefficient. So far so good. But why do we also have a distribution coefficient, and what is it, I hear you ask? Good questions. It's because some compounds, such as acids, bases and salts, can ionize in water, and this has a big impact on partitioning because ionized drugs are completely insoluble in organic solvents. It might not be immediately obvious why this is a problem, so let me show you some examples to help explain. On the screen is a schematic of a mixture of oil and water. We will assume there are 50 millilitres of each phase, and we have added 100 milligrams of compound. The final amounts of compound in each phase at equilibrium are 66.7 milligrams in oil and 33.3 milligrams in water. This means the concentration in oil is 1.33 milligrams per mil, and in water is 0.67 milligrams per mil. The ratio of these two is the partition coefficient. Imagine, however, that the compound was able to ionize in water. We might end up with different amounts of compound in the two phases, as shown on the screen. Now we have 40 milligrams of compound in oil and 60 milligrams of compound in water, of which 20 milligrams is unionized and 40 milligrams is ionized. When we measure the concentrations, say with UV spectroscopy, we cannot distinguish between ionized and unionized solute. So we will calculate the concentrations as 0.8 milligrams per mil in oil and 1.2 milligrams per mil in water. The ratio is now 0.67, not 2. The compound appears to be hydrophilic because the ratio is less than 1. But in reality, its affinity for the two phases is the same as it was before. It's just that we have calculated a different ratio because some of the compound has ionized in the water phase, and so it can no longer distribute into the oil phase. To account for this difference, we say that the experimentally determined ratio for compounds that ionize is a distribution coefficient, not a partition coefficient. You might notice, incidentally, that in the example I'm using, if we were to calculate the concentrations in the oil and water phases using only the unionized fraction, we could have concentrations of 0.8 milligrams per mil in oil, it's the same as it was before, as nothing has changed, and 0.4 milligrams per mil in water. The ratio is 2, and this is the partition coefficient, as it reflects the true relative affinities for the compound between the phases. Phew, that's complicated. So let me summarise. A partition coefficient represents the relative affinities for a compound between oil and water phases. A distribution coefficient represents the experimentally measured ratios of concentrations for a compound between oil and water phases, but does not necessarily reflect the true ratio of affinities because it includes a portion of the sample that cannot partition. If a compound cannot ionize, then the partition coefficient and the distribution coefficient will become equal. One final point to note is that the distribution coefficient is equal to the partition coefficient multiplied by the fraction of drug that is unionized. Right, that's all we need to know. A partition coefficient represents the true relative affinities for a compound between oil and water phases, while a distribution coefficient takes account of ionization. And where there is no ionization, partition coefficients and distribution coefficients are the same. I hope you found that description useful. If you did, please hit the like button and consider subscribing. It really helps the channel. Otherwise, thank you so much for watching. I'll see you again soon.